All right, you guys, welcome to the new unit, and we are going to cover genetics. So we're actually going to cover it in two lectures. So we're going to talk about the patterns of inheritance 1 and patterns of inheritance 2. So now that we've talked a lot about chromosomes and genes and homologous chromosomes, we're going to talk about how those genes are passed on to further generations. So we said that you get one gene from your mom, one gene from your dad, and they can be different. So how does that happen, right? And which one do you actually express? So that's what we're going to be talking about today. So what are we talking about today? So we're going to review a little bit about our genes, and we're going to talk about alleles. So what are alleles in relation to a gene? And then we're going to get into Mendel and his laws of genetics. He's really considered the father of genetics, and what he actually discovered with his research of garden peas. And then we're going to talk about the Punnett Square. How do we predict these ratios of genotypes and phenotypes? And then we're going to talk a little bit about monogenetic inheritance, meaning one gene actually gives rise to one trait. Okay, so that's what we're going to cover today, and then we're going to finish genetics in our next lecture. Okay? So let's learn a little bit more about Mendel. So he grew up kind of in what is now today the Czech Republic during the mid-1800s. And he actually was an Austrian monk who then decided to go to university, specifically the University of Vienna, where he studied plant breeding and mathematics. So what he found was he actually decided to work with garden peas and kind of what we may call them sweet peas nowadays. They look very similar. And why he wanted to study these was because they had a very short generation time, so he could really see how the genes were passed from generation to generation. They produced a lot of offspring, and you could really distinguish between the varieties. So they had really this very perfect flower as well, because they could either self-pollinate, so they could make a replica of themselves essentially, or they could be cross-pollinated. So that's just showing how he would cross-pollinate. He would actually cut the carpels off of one of the parent peas and then use the pollen from another one to pollinate. So that's how he cross-pollinated. So really they were a very easy plant to work with for genetics, because then he was able to see, hey, if I cross this, what do I get? And therefore he then kind of discovered these laws of genetics. So Mendel could really manipulate the outcome of traits. So he could see, okay, well, this flower is purple, that flower is white. What happens if I cross them? Do I get all purple? Do I get all white? And he figured out, if he did this over and over again, he could predict the outcomes of these traits and when he crossed certain things together. Okay, so he then could obtain very specific results, and then he was able to create the laws of inheritance. So this is just showing a few of the characteristics or the traits on the left that he studied, and what was dominant and what was recessive. So by breeding them over and over again and seeing the different outcomes from the crosses, he was able to see, what became dominant, what turned out the most, and what was recessive. So, and we'll go through these in a little bit more detail, okay? So before we get into Mendel's laws, let's talk first a little bit about alleles, okay? So we know what genes are, right? So genes code for certain traits. right? And so we said that a homologous pair of chromosomes has the same gene in the same location, but we said that could be a different, it could code for a different part of the trait, right? So we said, say it's hair color, we could say blonde or brown hair, right? So they don't have to be identical, and we call those alleles. Okay, so for example, you have a trait such as flower color, so if we're talking about garden peas, and then our alleles might be purple and white. Okay, so how do these guys interact? And for each organism, we carry two alleles, right? One allele from mom, one allele from dad, and that's how we get those homologous pairs of chromosomes, okay? So we said they don't have to be identical, but they could be identical, right? So say both your parents are blonde and they both give you a blonde allele for hair color, right? That's called a homozygous alleles, okay? So these guys are identical alleles. So that's our example on the left. So if we look at our we have two pairs of homologous chromosomes. On the left, we have one that codes for big B and big B. So those are the alleles for red and red, right? So they both code for the color red, and so they're homozygous alleles, okay? Same with our other one on right next to it on the right, and we have a little b allele on both chromosomes, and so they code for the color yellow. Okay, so yellow and yellow. Okay, so now what happens if they're not the same? So if we have different alleles, they're called heterozygous alleles instead of homozygous alleles, okay? So that's our example on the right-hand side. So our first pair of homologous chromosomes, one has a big B, which is red, and the other one has a little b, which is yellow. They both code for the same trait for a certain color of something, but they are different alleles, right? So same with our example on the right, it's just yellow and red, or little b and big B, okay? And then we'll talk about why we use uppercase and lowercase letters for these alleles, okay? All right, so we know that homologous chromosomes are going to share the same genes on the same loci, which is plural for locus, right? So we said that they don't have to be identical. They could be. So if they're identical, they're homozygous alleles. If they're different, they're heterozygous alleles. So this is just showing a homologous pair of chromosomes here. And we have 3 genes at 3 different loci. Okay? And so the first one is going to be homozygous, right? Because we have big P, big P. And then we have another homozygous allele pair, little a. And then at the end, we have a heterozygous allele pair, and that's big B, little b. So we said, what does the big letter versus the little letter mean? Well, the big letter means that that allele is dominant over the little allele, okay, or over the recessive allele, which is the little letter, okay? So that means that when this trait comes out, it's going to express that dominant allele. So say, you know, B stood for green, right? And little b was for yellow. Since the big B, green, is dominant, it'll be green, right? Versus yellow. So that's what we're going to talk about next, is the law of dominance in a little bit. So just to give you an idea of why we talk about big letters and little letters. So here is one of Mendel's laws, and this is the law of dominance. So now that we just said that the big letter is going to be the dominant allele and the little letter is going to be the recessive allele, what does that actually mean? So when we have a pair of alleles, if one is recessive, that means it's going to be masked or hidden, and one is going to be expressed or dominant. Okay, so if it's dominant, that's the one that you're going to see out in the trait. Whereas if it's recessive, you won't see it. You won't even know that they have that allele. Okay. And so when we talk about our alleles and genes, we're talking about a genotype. Okay. Versus when we're talking about the actual trait and the physical appearance of something, that's going to be the phenotype. Okay, and so the phenotype is always going to express that dominant allele, the dominant trait. Okay, and so only if you see the genotype will you actually realize, hey, there's a recessive allele there being masked. Okay, so if we look at our example on the right hand side here we're talking about peas again. So we have the flower color is going to be our trait, right? So if we're talking about purple color and white color, we say that purple is dominant over white. Okay, so let's start with our genotypes on the right hand side. Okay, so we have a homozygous pair of alleles, big P, so that means that it's going to be purple, right? So that's that dominant allele. Now the two in the middle are heterozygous alleles because they have a big P and a little p, but which one's dominant? Big P, which is purple. So they're both going to be purple even though they have that little recessive white allele. Okay, and the only time you're going to see a recessive trait come through is if it is homozygous recessive, and that's going to be little p, because then you can see your white flower. Okay, don't worry too much about these ratios right now. We'll talk about that when we talk about actually crossing different pairs. Okay. So another law is the law of segregation. And this just kind of makes sense. We've already kind of explained the law of segregation when we were talking about meiosis, but when we talk about two alleles of a single trait, they're going to separate randomly, right? So when they separate, it's a 50% chance that either allele will end up in either gamete, right? It doesn't mean that this gamete will receive this allele and that there's a 50% chance either way, right? So say you have big H, little h, right, on the right hand side, there's a 50% chance you're going to get a big H in there and a 50% chance you're going to get a little h. And it's pretty simple, right? So essentially you have, two copies of the gene, they're going to separate and end up in different gametes, right? And then we've already talked about the law of independent assortment. So genes do not influence each other during meiosis, okay? That means that alleles are sorted independently of each other, okay? So that means if you've got, a gene A here and a gene B, they're going to independently assort into those gametes. There's every possible combination that could occur, and there's an equal probability that you will get the different combinations, right? So if we talked about these two genes on different homologous pairs of chromosomes, you're going to get 4 different outcomes from those pairing, right? So we've already talked a little bit about independent assortment during meiosis, right? So that's just showing you, again, how we get variation, and there's an equal probability or possibility of getting any one of these 4 gametes, okay? So now, what happens if we cross these different genotypes? What kind of phenotypes and genotypes are you going to get in each generation? So Mendel came up with a way to predict these different ratios of what kind of genotypes and phenotypes you're going to get if you cross these different patterns, these different genes together. And he uses what's called the Punnett Square. So I'm sure if you guys have had any sort of biology before, you may be familiar with the Punnett Square. And so essentially it's going to predict the ratios of each phenotype and genotype in the next generation. So if we have an example of our flower color, our alleles are going to be purple and white, and we said that purple is dominant, white is recessive. So what are our possible genotypes, right? So our possible genotypes are big B, right? A homozygous dominant, which is going to be purple, so that's our phenotype. And then we also have a heterozygous, which could be big B, little b, which is also going to be purple phenotype. And we could get a homozygous recessive allele pair, little b, which will give us our other phenotype of white, right? So these are our possible outcomes if we cross this trait together, right? If we cross these traits. But what happens if you cross specific plants, right? So that's what we're going to talk about and how the Punnett Square helps us. So let's do some examples. So this would be considered a parent generation. Okay, so this would be a homozygous dominant purple plant. Okay, so big B, and we're going to cross it with a homozygous recessive white or little b, right? So if we see here, F1 means parent generation. So this is an F1 cross. So here's our white across the top and our purple to the left on the side, right? Now we said because of our laws of segregation, it's a 50% chance whether you're going to get either allele, right? So we have to go through the options of whether you're going to get a big B or a little b. Now these are easy because they're homozygous, right? So either way, you're going to get a big B from the purple on the left, right? So we know we're going to get a big B in all 4 squares, right? Because he's going to always throw a big B either way, right? And same with white. You know that white is always going to throw a little b because there's no other option, right? So that means that our child generation, our cross between these two parents, will always give you this heterozygous big B, little b. But what's the phenotype of all of them? That's right, they're all going to be purple, right? So they're all purple because of that dominant B, okay? So here are the answers to our last cross, right? So our parent cross creates the F1 generation. So what is our genotype ratio? Well, we won't have any big B or little b. We have all 4 big B, little b's. Okay, so that's our genotype ratio. And then if we talk about our phenotype ratio, we're only talking about purple and white are our 2 phenotypes. And we have all 4 purple, right? Due to that purple dominant allele. Okay, so now what happens if we cross 2 from these F1 generation? We're going to call it an F2 cross because now we're going to get the next generation. So again, we have 50-50 chance of getting a big B or a little b, so they're both heterozygous. So now how do we fill out our cross? So essentially you're going to go this way across with both of your alleles, right? And then you're going to go top to bottom with both alleles, okay? So if we bring our big B over here, it's going to be in both, right? So big B, because that's the only allele here that we can have. And then we have our other allele, which is little b, right? And now we go to our other F1 generation, which is also heterozygous. So we bring those down. So we have big B here, and we bring down little b and little b, okay? So now we have maybe something a little more exciting, right? So we have one big B, and we have two heterozygous big B, little b's, and we have one little b, okay? So what are our phenotypes? Well, we have 3 purple, right? Because we have 3 big B's, and we have 1 white. So here is our outcome for that F2 generation. So we have our genotype ratio and phenotype ratio. So 1 to 1, right? So 1 of the big B, 2 of the big B, little b's. And then 1 of our little b's. That's 1 to 1 ratio. And then our phenotype ratio is 3 to 1. So we have 3 purples to 1 white. So we finally get a white, and that's that recessive trait coming through because we finally have a homozygous allele pair, right? So that's our F2 generation. So now Mendel also tried looking at 2 traits at the same time on different chromosomes. So he wanted to see, let's say, seed color and seed shape and see what would happen. So he would cross and look at those 2 at the same time. And this is really where the law of independent assortment is revealed, because you're tracking 2 characters at once, and that trait is on 2 different chromosomes, right? 2 different homologous chromosomes. So if we follow seed color, we can either have yellow or green. And then if we follow seed shape, it's either going to be round or wrinkled. So remember, if we have the law of independent assortment, there's an equal probability that these chromosomes are going to arrange 2 different ways, right? So you end up with a 4 different possibility, okay? So we call this a dihybrid cross, where you're looking at 2 traits at once. So essentially what happens is you have the parent generation, which is homozygous for each the dominant traits and then each the recessive traits, okay? So in this case, the big Y is going to be yellow and the little y is going to be green for the first trait. And then we have a big R for round, which is the dominant trait, and the little r for wrinkled, okay? So if we cross those, remember a parent generation is always going to give you the F1 generation of the heterozygous of each, right? So big Y, little y is always going to give you yellow, and then big R, little r is always going to give you round. So you're always going to get that same genotype and then that same phenotype, which is yellow and round, right? So now we're going to actually cross this F1 generation. So you're going to cross 2 heterozygous traits, right? So we actually have the 2 traits and they're both heterozygous, okay? So you can follow this through when, you know, it's quite complex down here in terms of what you're going to get for your F2 generation. So essentially what happens is that, remember, we have our 50-50 chance of each allele, right? So that means that just this one is either going to give you a YR gamete, a little y big R gamete, big Y little r gamete, or little y little r. So these are the 4 possibilities, right? We said independent assortment, we get 4 possibilities. So that is those are our 4 different gametes that we can get. Same with our other parent, right? So they're going to give you the same gametes because they're both the same heterozygous, okay? And then you just follow your way through, right? Just like we did before, across, and then you're going to go down. And you're going to end up with all of these different options, okay? But the big thing is to look at your phenotype ratio. So the phenotype ratio is really what I care about, okay? And it's always going to be the same. So you're always going to have 9 of the 2 dominant traits, so yellow and round. You're going to have 3 of each of the 2 different heterozygous traits. So you're going to have 3 that are actually green and round, and you're going to have 3 that are yellow and wrinkled. So those are your 2 kind of mismatched ones. And then you're going to have 1 that's your all recessive, so green and wrinkled. Okay? So just to review, if you have 2 parents that are heterozygous for both traits, regardless of the traits, what is your phenotype ratio? It's going to be your 9-3-3-1. So that's kind of the take-home message that I want you to get through for this dihybrid cross, okay? So we're just talking about this heterozygous for both traits cross. Now you can work your way through this entire Punnett Square just to kind of prove to yourself those different numbers. But again, it's hard to do on the computer, but I can go through it with you as well. If you want to set up any sort of meeting or in office hours, I can go through it, okay? So now it's time for a little bit of a review. So go ahead and pause your video and go through these different definitions, right? So what is the difference between a genotype and a phenotype? What's the difference between a dominant versus a recessive allele? What is homozygous versus heterozygous? What is an allele versus a locus, right? So go ahead and review those. And then work your way through this little question down here. And essentially it's talking about if an individual is heterozygous, big B, little b for a gene, according to the law of segregation, each gamete formed by this individual will have either a blank allele or a blank allele. And then we're going to talk a little bit about which step in meiosis is this the physical basis for the law of segregation. So go ahead and kind of think back to meiosis and how this law of segregation actually is happening. Okay, so hopefully you were able to pause the video, go through those definitions, which I'm not going to because there's quite a lot of them, but you do need to know all of these definitions. But let's go through our little review down here, right? So if we have a heterozygous gene, big B, little b, each gamete is going to either have a big B allele or a little b allele, right? And then what step in meiosis do you think this law of segregation has the physical basis for? Okay. It's metaphase 1. So we talked about, meiosis 1 versus meiosis 2, and so I have a little explanation on the next slide for us. So when we're talking about the law of segregation, we're actually talking about what's happening during meiosis, okay? So it's metaphase 1. Right? So if we have our 2 homologous chromosomes that are set up, right? One is going to go into one allele and one is going to go into the other allele. And it's a 50-50 chance, right? If you're going to get the blue chromosome or the red chromosome. And so this is pretty crazy that, Mendel didn't even know about DNA or genes because we hadn't really discovered them yet. So this is pretty crazy that he really understood the law of segregation and what is happening in our chromosomes and in our genes without even understanding what a gene was or what DNA is, right? So pretty crazy. So a lot of inherited traits in humans are controlled by multiple genes, but there are some that are controlled by a single gene, and we call that monogenetic inheritance. So one gene can affect an entire trait for that human or for all of us. So some examples of that are albinism. It's where an enzyme that produces pigmentation doesn't function properly, so you have reduced pigmentation in your skin and you become an albino. And this is a recessive trait, okay? Now, freckles, which is a dominant trait, is also one single gene that gives people freckles, and freckles are actually dominant. So now we might say, okay, well, if we see things in how common are these traits in a human population, right? So dominance does not always necessarily reflect the popularity of that gene or the commonality of that gene. Okay, so if we look at, say, pigmentation, right, that makes sense because normal pigmentation is the dominant trait, whereas albinism is the recessive trait. So that makes sense because we see this less common trait, and the less common trait is a mutant trait. Okay, and the more common trait is a wild type trait. Okay. So when we say wild type, that's the most common in the population. So that one fits, right? Normal pigmentation is the wild type, but it's also dominant, okay? Whereas when we look at freckles, freckles is a dominant trait, but it's not the wild type because it's not the most common in a population. It is actually the mutant trait, even though it's dominant. So the recessive trait of having no freckles is actually the wild type, okay, because it's what we see in the population. So again, dominance has no bearing on what's most common in a population. I mean, usually you might see the dominance, but again, it's not always true. So that's why we have different terms where we say a trait is a wild type trait, which is more common, and then a trait that is mutant, which is less common. And they can either be dominant or recessive. So here are some more examples of inherited traits that are controlled by a single gene. So these are some genetic disorders, and some are inherited as dominant or recessive traits by a single gene. But again, it doesn't tell you whether it's wild type or mutant. Usually when we're talking about genetic disorders, we're talking about mutant traits because they're not as common. But if you notice, there are some dominant disorders, such as Huntington's disease, achondroplasia, which is part of dwarfism. So again, you see these recessive and dominant disorders, which are only caused by a single gene. Okay, so again, in the incidence on the right-hand side, you see are fairly low. Maybe in certain populations, they might be a little bit higher than others. So just kind of interesting to think about where, you know, some traits are caused only by a single gene and they could be dominant or recessive. So let's kind of come back to Mendel. He presented his research about 7 years after Darwin's Origin of Species. And then he didn't get a lot of acknowledgement during the time of his research, and he actually died before he was acknowledged for his work. So he died in 1884, and he wasn't acknowledged until the 1900s, where 3 different scientists actually independently duplicated his work and rediscovered his writings and said, hey, if this guy was actually right, let's give him some credit for it. So again, it's kind of one of those sad stories where, you know, a scientist. This happens with artists and things like that too, that, you know, they're not really acknowledged for their work until a lot later. You know, we saw a little bit with that with Darwin. He was kind of too scared to, you know, publish his research and his findings and share his findings with the world because they were so against what was happening at the time. And so Mendel did present his research during his life, but he wasn't acknowledged until after his death. So we're going to finish up with some practice. So you guys are going to do this at home. So you're going to pick two, you can pick one character or two characters, and you're going to cross two heterozygotes. So that's going to be your first Punnett square. So you're going to pick a trait, cross two heterozygotes, meaning they're each going to have one dominant allele and one recessive allele, and see what you get. We already did a cross of two heterozygotes, so that should help you out. And then you're going to cross a homozygote and a heterozygote. So we haven't done that yet, so that should be interesting to see what you get. And you can either do the same trait or a different trait for the second Punnett square. So that should help us out with some practice, and then we will have a worksheet on genetics as well next week. Okay, so we'll finish up genetics with our second lecture next week, and it'll be really what happens if we change Mendel's laws and if they don't always apply. So what happens if two things are, you know, both dominant, or what happens if we get a blending of a dominant and a recessive trait. So we'll talk a little bit more about that next week, as well as some sex-linked traits. Okay, so that's what we'll do next week. All right, thanks a lot. If you have any questions, let me know.